Our speakers today will be Sandra Rodriguez-Cruz from the DEA and Sarah Kearns from the FDA. And we're going to start off with Sandra. So Sandra, feel free to share your screen and tell us how you guys are using DART at the DEA.
All right. Well, thank you very much. Good afternoon for those of you on the East Coast. Good morning on the West Coast. Thank you again for the invitation to participate during this course. And yeah, I'm going to be telling you guys the story of how we've been going about implementing, validating and the use of DART-MS, the most recent use of DART-MS throughout the DEA laboratory system. I'll start with two short disclaimers in terms of the opinions I will be expressing do not represent the views of the DEA or the Department of Justice or the United States federal government, as well that I'm going to be mentioning some commercial products, and that does not constitute approval, endorsement, or recommendation by DEA, DOJ, or the U.S. Federal government. I'm going to separate the presentation into three different sections. Most of it is going to be concentrated on the second section, but I will take you through a little bit of the history and some of the lessons that we have learned in the past when we have tried to implement some of these ambient ionization mass spectrometry techniques. Then I'll go and try to give you as much of the story of what we have been working on for the last 2 years in terms of standardizing the use of DART-MS throughout the DEA labs. Everything from procurement, evaluation of the new instruments, what we did for method validation, development and validation, all the policy and procedures that we needed to take care of, go-bys, on-site training, as well as the method verifications that the laboratories were assigned. And I'll finish up with the future directions. So the previous use of ambient ionization in the DEA lab has been a little bit tumultuous. We had, shortly after I came to the system back in 2002, between 2005 2014, we had 4 quadruple ion trap systems that were equipped. They were LC-MS systems, but we also did desorption, electrospray ionization. So we had 4 regional laboratories where these got intermittent use. At that point, only 4 of those where we could switch between the LC-MS configuration and the DESI-MS configuration. After that, and those were on low resolution quadruple ion trap systems. Later on, after that, there were 2 regional laboratories that incorporated the use of DART-MS on both linear trap as well as orbit trap system. In the San Diego lab where I was working up until 2019, in 2019 we obtained another LC-MS system, also a low resolution quadruple ion trap, that we could also switch between LC-MS configuration and DART from the ion sense sources. And after that, we also had, at the same time as that was happening, actually, we had 3 regional laboratories that had a QTOF system kind of from the past. They've had it for a while, and then we purchased DART sources back in 2022, so we could have a dual purpose on those systems as well. We had some, hurdles and a lot of lessons that we have learned throughout all this time. One of the main impediments was, the lack of standardization. Not all laboratories using the same system. Our LC-MS systems also under the umbrella of our procurement processes, they were considered a research instrument, and that certainly resulted in some hesitation on anybody wanting to go and try use the instrument and learn how to use it. A lot of folks were afraid to break the instrument with the worry that they were going to be breaking a research instrument. So not a lot of use, limited access also to the instrument and expertise. For the most part, there was just 1 instrument per laboratory. So this ended up limiting the use of the laboratories. We always had some folks and chemists in different labs that would take up the initiative of learning more about the instrument, taking the time to learn and to use it consistently and proficiently. But those were definitely more the exception rather than the rule. Now, as of 2019, we're trying to make meaningful changes throughout the laboratory, not only from the point of view of ambient ionization, but also from the point of view of method development and validation. And central to that, to those changes, has been the formation of our method development and technology transfer group at the Special Testing and Research Laboratory. And that's the group that I've been working with, where not only for DART, but also for our GCMS methods, LCMS methods, GCFID, IR methods, all those validations that used to happen in-house in all the laboratories are now done here. We develop the method, we design the method, we validate them, and then we transfer the method to the regional labs where they undergo some method verification. And that way, that's how we have been performing standardization, system-wide standardization through our laboratories for the better part of the last 4 or 5 years. We have also gotten into the mode of providing customized in-house training, either virtually or in person throughout the laboratories. And as part of some of these changes, we also want to try to allow analysts and managers to lead their chemists, time to learn the new technique, which is a little bit difficult often to achieve, but we're going to keep trying on that end. In terms of standardizing how we are going to be using DART MS throughout the DEA laboratory system, I'll take you through this process. For us, this is being encompassed in 2 phases. During the fiscal year 2023, we purchased 5 different instruments. The first instrument came here to the Special Testing and Research Lab, and the other 4 instruments went to our New York lab, our Miami lab, Texas, Dallas, Texas, as well as our Southern California lab in San Diego. And later on in fiscal year 2024, which just started, we'll have phase 2, where the same instrument is going to be installed in our Maryland laboratory in Largo, Chicago, Illinois, as well as their Northern California laboratory in Pleasanton up in the Bay Area. And in addition to that, we're also going to be installing an instrument at our DEA Training Academy in Quantico, Virginia, so we can start training folks as they go through our basic forensic chemist class. As I take you through this process, I'm going to go through how we evaluated these instruments, how we selected them, the delivery and installation, how did that work with having 2 vendors, 1 vendor for the LCMS, 1 vendor for the DART. We also did some 3-day on-site LCMS training that was provided by the commercial vendors. This was done in all the laboratories. And what we did for DART MS method development, standardization, and validation. And I just finished going through the 4 sites, providing a 3-day on-site training on DART, and I'll show you what we did on those and what we do for method verification. And after that, and now we're starting the phase of developing methods in LCMS to then standardize them and implement them throughout the system. Our future vision, and we are very somewhat optimistic, though aspirational, is to eventually have 3 DART MS systems in the laboratories. Usually our field laboratories are divided into 3 analytical groups. So to have 3 DART MS systems, 2 of those will be dedicated DART MS systems. And the third one will be a dual configuration system where we can switch between DART MS and LCMS. We don't have as much need for LCMS as we're going to have for DART, so we don't really need 3 LCMS systems, but the 3 DART MS system will definitely come in handy. This is a timeline that gives you a general overview of what we have been working on since the end of calendar year 2021, which was the first quarter of 2022, where we started talking with vendors, doing demos, evaluating instruments. Lucky for us, the time during COVID was a time where all these vendors got really good at providing virtual demonstrations. So we didn't really need to go and travel to these sites. We were able to do a lot of this on virtual meetings and virtual discussions. And then we spent the better part of that quarter just putting together specifications and going through the procurement process. And then we waited for the systems to be shipped and delivered. Once that happened, we needed to schedule installations. I'm using the numerical denominations here that we use for our different laboratories, the SFL1 being the Special Testing and Research Lab. Our laboratory number 2 is New York, laboratory number 4 is Miami, and laboratory number 8 is San Diego. For this phase 1, we were working with the even number laboratories. Phase 2 is going to be the odd number laboratories. So different schedules for the installation, not only the installation, but also the on-site thermo training. And I'm going to go through when did this happen, but as you can see in the timeline, having to schedule and coordinate not only the installations, but also the on-site training, then the installation of the DART, and then our visits from our method development and technology transfer group to provide the in-house training at the different labs. That brought us up to about 3 weeks ago when I visited our San Diego lab to provide the training. We also put together a lot of the deliverables. I'll give you some screenshots of some of what we produced to facilitate this implementation. Not only we developed the method, standardized it, and created our validation packet, but we also created multiple go-bys to guide analysts into how to switch from LC-MS to DART-MS and vice versa, as well as the step-by-step go-by for actually doing DART-MS analysis, starting from preparation of the solution, how to revise the method, and depending on what your target compound of interest is, how is it that you can revise your DART-MS method in order to isolate the target ion of interest, data processing. All right, and so different go-bys, everything presented in green were deliverables that came from our group. During the method, the vendor evaluation and selection, there were some important requirements that we were looking for. We were looking for a system that was rugged, a system that was easy to use, easy to maintain, as low maintenance as possible, a reasonable footprint. We don't have excess space in the laboratories, and if eventually we're going to put 3 of these systems in each group, in each laboratory, we wanted this to be easily accommodated on the simple basic benchtops that we had, and we also needed to consider cost, given the fact that we were going to buy 4 or 5 instruments the first year and then 4 on the second. One of the first alternatives we did consider was the quadruple ion trap model, since those were relatively cheap and very robust based on the experience that we had with them before. But as we learned, that were no longer being manufactured. So we went on to the next one in considering the triple quadruple systems, and systems that are demonstrated to be very robust in toxicology laboratories and relatively low maintenance. We evaluated 4 vendors, and like I said, we had a lot of virtual meetings and demonstrations. This is an example of a spreadsheet that we kept every time we did a demonstration. They did a demonstration for us in the discussions that we had. We evaluated 2 systems from Agilent, 1 from Shimadzu, the system from Thermo. And then the merger, the purchase of ionization by Bruker was kind of happening at the same time. we did not know, IonSense had let us know that they were in conversations with the vendor. We didn't learn until multiple months after that vendor was broker, but we did get to take a look at what they were going to be putting out. In order to facilitate this procurement within our procedures here within DEA, we created a specification sheet that then we forwarded to our procurement and contracting. They put it out for bid, and we didn't hear for a while in terms of which instrument we were going to get. And then one day we were told that, all right, you're getting a thermal triple quad, and this is what the system looks like. It's a benchtop instrument, relatively small footprint, with an electrospray source. This is what it looks like, the actual LC-MS system. Behind this bench, we have an air compressor with our nitrogen generator, as well as the pump. We have significant large amount of space here at the special testing, so we were able to accommodate those pretty nicely. And then this is what the system looks like with the IonSense dart source installed in front of it. So we have a dual configuration system, and this is the same configuration that was as well installed, installed through the 4 regional laboratories. So in terms of deliveries of the LCMS system, it took between 3 and 4 months. The liquid chromatographs were coming from San Jose, the triple quads were coming from Germany. We actually were almost on time, and then we heard of a separate client that ordered 25 instruments. So we were bumped down the list on delivering an installation from the thermal point of view. And because of the variabilities in availability for engineers to install this instrument, it took about 4 months from the moment, from the installation of the system here to the last installation in the Miami laboratory. As soon as we received them and they were installed, the vendor does not do a formal LC-MS test and separation. So we had to come up with a standardized LC-MS checkout procedure with a mixture and a quick method that we could transfer to the laboratories that were receiving the instrument and getting the instrument installed, just so we could confirm that the instruments were operating, and so we could sign the bill and pay the manufacturer. And we were glad we did that, because there were some issues encountered. There was somewhat of a variability in terms of how different engineers in different regions of the country install the instruments. So it was a good thing to actually not let these instruments, confirm on the installation of them and release them until we checked that they were able to produce consistent results with the LC-MS checkout procedures that we delivered to them. And then somewhat overlapping this, but not completely simultaneously, we also had the delivery and the installation of the DART sources. IonSense, we had to ask them to wait until the LC-MS systems were installed, and then they came in and they installed. And that also took, the whole window took about 4 months as well. As I mentioned, we also had a 3-day on-site LC-MS training, and this was included in the bundle that we purchased. We got into conversations with the vendor, and we were able to make sure that the same trainer was the one that went to every laboratory. So that person came here first, ahead of going to the other laboratories. And here we did discuss and went into the deep, full capabilities of the triple quad system, a discussion about the non-routine approach that we were going to take with this instrument, because they're not used to being used on generating full MS data. Most of these instruments out there are used for quantitation, SRM and MRN experiments, and we were going to try to use this to collect not only full MS data, but also MSMS, where we can collect a full fragmentation spectrum, not just looking at some fragments, selected fragments in fragmentation channels. We went into the maintenance and calibration as well as some method development from the LCMS point of view, and the discussion on how master methods come, as well as some quantitation capabilities that we may go into the future. The extent of this discussion only happened here. So we discuss and customize so that the information that this trainer was going to provide to the other laboratories did not go to this extent, as we are going to be the ones developing the methods. They did not need to learn how to develop methods, LCMS, and all that. So because we requested the same trainer, and this trainer also had other clients, we also encountered some scheduling hurdles. And between, he came here at special testing in April, the last week of April, and it wasn't until the last week of September, actually the week before I went to San Diego, the second week of September, that he was able to do the training in our San Diego location. In terms of method development and standardization, like I said, everything happened here at the special testing and research lab. And for right now, we're just standardizing and validating the DART MS configuration, nothing on the LCMS side. And that was completed between May and June of 2023. We validated multiple variations, the use of the quick strip cards, as well as manual sampling. Our laboratory in New York actually added to that validation by working and validating the use of the DPIT samplers. And we just received a tablet module that we are going to be evaluating and validating for eventual future use, as well as implementation through the labs. In terms of MS analysis, we validated two methods. One for full scan with a scan range of 50 to 500 for presumptive analysis, to screen single or multi-unit samples and provide this molecular weight information. And we also validated a method, MSMS method, that could be customized depending on the target analyte that you're looking at, so that we can generate confirmatory data by providing a fragmentation pattern, and that way fulfilling the requirements of a sweet drug category A. These are the four method variations. The _QS and _M distinguishes the different variation in terms of sampling using the DART size. So this is where the four variations come from. Two of them are doing DART MS, and two of them are doing DART MSMS with both the quick strip as well as the manual sampling. For a method like this that we consider a confirmatory method, we do an extensive selectivity evaluation as well as an accuracy evaluation. Right here, I'm showing our master selectivity spreadsheet, where we list the compounds as we add them to the validation, the pseudomolecular ion that is observed. And if we observe ammoniated molecular ions, we also list them here. The collision energies that are evaluated, and for MSMS evaluation during the validation, we evaluated 8 different collision energies from 5 to 40 volts, as well as any limitations that we may encounter. And we also identified the optimum collision energy for each compound, and those are labeled in red and in bold. And we define that optimal collision energy as that energy where we can obtain the maximum fragmentation, but where we can still see some of the molecular ion. In terms of the accuracy evaluation, we compare at the special testing and research lab. When we do our validation, we compare it against a library that's external. So the NIST both low resolution and high resolution libraries come in handy. For this, so we reference our data and compare it to them, and we check for a general correspondence in terms of the fragmentation pattern relative to that reference spectrum. We check the ion abundances, M over Z values, as well as the isotopic distributions that we may see, and make sure that the prominent ions are the same nominal mass. Again, we're working with low resolution mass spectrometry on a triple quad. Make sure there's no unexplainable extraneous ions. Be able to explain and identify that the pseudomolecular ions are either the protonated ammoniated species and correspond to the theoretical mass of the compound. And like I said, multiple collision energies well evaluated and the optimal collision energy identified. And for a lot of these accuracy evaluation, we use the NIST MS search program, and we produce a data to support the validation that presents the data we obtain and then compares it with the NIST MSMS library. We can also do that internally within the freestyle software that comes with the thermo instrument. Again, with the upper spectrum corresponding to our analysis and the bottom spectrum, in this case, the low resolution MSMS NIST library. Now, in addition to the method validation, like I mentioned, we also needed to come up with those kind of ad hoc LCMS checkout procedures so we could sign up on the instrument. We also need to provide laboratories with performance verification procedures. These are the procedures, standardized procedures for their monthly checks, and how we make sure that the instruments are working up to par every month, as well as the routine maintenance procedures recommended not only by the manufacturer, but also by us. And we also created two libraries, a library for full scan data, pretty much a library of molecular ions, as well as MSMS library, internal library here. And these will be used by the laboratories in order for them to then verify the methods and compare it to us. Then we went on to train, and the 3-day training, this was the general schedule on the different laboratories. On day 1, we gave them an overview of the DART and LCMS system, as well as some background on soft ionization techniques, as well as MS and MSMS data. And then took them through the outline of how we were implementing this through the DART MS system. We discussed a little bit of also internal policy at this point. And during the afternoon of that first day, we go hands-on on the instrument. And during the first day, this is just limited to what we call the instrument monitors. These are the folks that do the routine maintenance on the instrument, the folks that would be doing the switching between LCMS and DART MS, as well as the members of our quality group, since those are the folks that will be doing the monthly performance verifications. And we took that afternoon to go through all the go-bys that we prepare so that they would be comfortable with all the procedures that they were going to be asked to do. For the complete day on day 2, as well as the morning of day 2, then we did 2-hour sessions with 3 to 5 chemists at a time, where we gave the opportunity to every chemist to actually drive the instrument, perform the analysis, sample the quick strip, do manual analysis, as well as go through the go-bys, print data, review data, process it, and also do library searching. And during the afternoon of Friday, as part of introducing this technique to the laboratory system, then we needed to do some competency testing. So before I left the building, I competency tested 2 to 3 analysts, and either instrument monitors or members of the quality group, and then they would go and continue that competency testing with their colleagues at their respective laboratories. This is a screenshot of the go-by that we provided to them in switching from DART MS to LCMS, as well as from LCMS to DART MS. Step by step, starting from, disconnecting the black hose from the flange, and ending with every, performing a verification procedure. Every time we end up in a new configuration, they need to do a performance check and document it to make sure that all the cables and all the lines are connected and the instrument is acquiring data appropriately. This is a screenshot of the performance verification template that we provided them. This is a pretty standardized format that we use for all of our techniques. And here we provide them with instructions on what to do and how frequently to do it, the mass spectrometer calibration, as well as the data mass performance verification. We're providing them with a recipe that they need to use for the performance sample, as well as the acceptance criteria, which is just exported out of our policy manual. This is an example of what that performance mixture looks like for Dart MS. For separation techniques such as chromatographic separation techniques, we usually use performance verification mixtures that contain an early eluter, a middle eluter, and a late eluter. And we did the analogous for checking the performance of a Dart system. This is for the full scan mode. So we selected methamphetamine as a low molecular weight ion, dextromethorphan and diltiazem, so that we can ensure that the right nominal mass and all the clean molecular ions were observed. And we also asked them to collect MSMS data for each one of them. And this shows you one of the printouts that can be observed. We divide the chronogram into the 3 different analyses that had been done: MSMS on methamphetamine, MSMS on dextromethorphan on the second panel, and MSMS on diltiazem. And that's the check that they will have to do after they switch into Dart MS mode. This is what the go-by looks like for doing Dart MS analysis, as well as editing Dart MSMS method. And like I said, it starts by asking them to prepare their solutions, a suggested concentration, how to spot the samples, all the way to analyzing the data. And here we tell them how to create that customized MSMS method. And we also told them how to process the data using the software from Thermo. In this case, we went with the Qual browser. And some of them, because we had Thermo instruments before, some of them were familiar with it. We provided them with some standardized templates that they could use either in portrait mode, like I'm showing here, or in landscape mode. And the information contained on the top of the printout fulfills all of our policy requirements in terms of, the instrument that was used, the method that was used, all the minimum method and acquisition conditions, as well as this is, the data path, the date and time of analysis, as well as some of the unique identifier information for the samples that will be received in the laboratory. And we also show them how to do library searches and how to print out a library. And the way we train them to create the libraries is so that each library entry also at the bottom will have all that traceability information that we need in order to be compliant with our policy in terms of which instrument was used to collect the MSMS data, the name of the reference material, the collision energy that was used, the lot number, as well as the date that that reference material was tested in that particular instrument. So we were able to customize it and create the go-by in order for them to do that. And this is what that go-by looks like. This becomes, once they create their in-house library, this becomes their library of positive controls. In order to make a positive identification on a confirmatory test, analysts are required to compare it to a reference material that had been tested under the same conditions in that same instrument. So it is favorable for us to create these in-house libraries per instrument so that we can just, by doing a library search, we quickly fulfill this requirement. And as they form these libraries, this can be used and they grow with the age of the instrument. And these positive controls only need to be tested once in the lifetime of each instrument. For this, on creating the in-house library, we also use the NIST, the capabilities of the NIST MS Search program. And again, we teach them how to become the librarian and how to generate the information to fill out, all the comments on the library entries. Method verification, which is a procedure that the different laboratories that are receiving the method would have to do. And they do limited work, not as extensive as the one that we do during the validation. And a lot of the data can in fact be collected while we're doing the on-site training, if we have time, or shortly after. At minimum, we want them to do a selectivity study with at least 8 compounds. A lot of them usually do more than this. And an accuracy evaluation using both methods, standardized, the DART-MS and the DART-MSMS, and at least 2 of the sampling variations, meaning either the quick strip cards or the manual sampling. We don't ask them to do all 8 collision energy levels, just one, the one that we determined that was the optimized collision energy. And what we ask is for them to take that fragmentation data and compare it to the data we obtain at the Special Testing and Research Lab, so that way we kind of establish the traceability. We compare to an external source, a NIST database, and they compare against us. And this will be required for all laboratories receiving the methods. Now we will use the data of the first 2 labs to supplement the original validation. When we create a validation packet, it's formed by not only the report, but also that master selectivity list, which is a living document that we keep on an Excel spreadsheet, the validation data that we collect here, and then the verification data that 2 of the labs collect. And that forms the ruggedness and reproducibility evaluation part of the packet. So as of the end of 2023, which was just last week, we have the validations completed for those 2 methods, the one that produces presumptive information, the one that produces confirmatory information, as well as develop MS and MS libraries. So these are being developed, but again, growing as we add more reference materials to them. Completed the on-site training at 4 of the laboratories. The verification was completed at 2 of the labs for that ruggedness assessment, and that validation report now has been officially posted and made available now within our document control system. And the verifications on the other 2 laboratories are ongoing. We have competency tested over 10 analysts, and more are in progress, since each of the folks that I competency tested, now they are competency testing their colleagues at their respective labs. And while this is all happening, we also the purchasing of the additional 4 units that are going to our odd number laboratories in Miami, Chicago, and the Northern California laboratory, as well as our training facility in Quantico. Those are being procured at this time. We also participated in the NIST ambient ionization mass spectrometry interlab study that happened during the summer. And I had some examples of spectra that I took around with me during the on-site training. On the upper left is a chromatogram of one of the samples. This was NIST number 17, containing 2 components. And I like to compare and contrast this. That was run as one of our standardized methods, GC high. That takes about 2 minutes. It's quite a fast method, but then we can get the same information, 2 components, molecular weight information. And the only difference is that this DART-MS analysis took only 12 seconds to acquire. Similar with this sample that had 4 components. On our GC high, it took 4 minutes. We'll see 4 peaks. If we do DART-MS on a 12-second method, we can also see those 4 components. When we analyze the mixture that contained 3 components on this 10-minute method, containing the methamphetamine, benzyl, fentanyl, acetyl fentanyl, we saw 3 peaks. In the chromatogram, But then in the DART, we only see 2. Why? Because acetyl fentanyl and benzyl fentanyl are isobaric. They have the same molecular weight, but once we do MS fragmentation data, we can see that the spectra we are actually getting is a combination of both the acetyl fentanyl and the benzyl fentanyl, and that by doing the library search, that could be easily assessed. In terms of future directions, now we are moving on into the incorporation, the official incorporation of DART-MS as one of the options, one of the tools in the toolbox that our analysts can use, and our incorporation into our own analytical schemes. It is already an option on a SOP that we have for analysis of suspected opiates. And the next one that's going to be incorporated into is our standard operating procedure for methamphetamine analysis. We also have some ongoing short-lived projects to kind of supplement and inform our analysis of the isobaric compounds that where DART can be used to distinguish, DART MSMS can be used to distinguish or not distinguish positional anisomers or other isobaric compounds, as well as we are doing also some analysis on thermally labile compounds, some that we cannot not see with DART, and some that we in fact can see with DART-MS, and we're optimizing those temperature conditions of the DART source in order to optimize for that. At the same time, we're also developing and validating LCMS methods. So that's kind of a parallel channel that we have, and we will be providing also LCMS training and implement those methods throughout the lab. And once the second phase of instruments is delivered and installed, then we'll start our on-site training all over again, and into our other regional laboratories, the 3 ones that we have left, as well as the DEA Training Academy. And we will do just what we did in the 4 laboratories during the last quarter. We will repeat for the 3 laboratories in the academy, training academy that's left. And that will be it. Be happy to take any questions.
Okay, so thank you, Ed, for setting up this fabulous workshop and for giving me the chance to participate. Every time I get an email from Ed, I get excited because I know something fun is about to happen. My name is Sarah Kern, and I've been an analyst at the Forensic Chemistry Center of the Food and Drug Administration for the last 15 years, 12 of which I've been focused a lot on DART-MS, so it's certainly my favorite technique. In July, I changed positions to be the supervisor of our satellite laboratory operations group. So I'm still kind of getting my feet under me in that role, but it's been very exciting. And I'm happy to share with you today some of the work that we've been doing with that project, which is now a program. So it's certainly been a beautiful collaboration. You can see here on this slide that many of the analysts that worked on this program are from the Forensic Chemistry Center, or FCC. But you can also see that there are lots of analysts from our pharmaceutical labs, our medical products labs, and our specialty products. So it's been a wonderful collaboration across the entire nation. And I do also have to do my disclaimer because I am not an official spokesperson for the U.S. Food and Drug Administration. So I'm certainly not promoting any particular products or companies. Okay. International mail facilities and the FDA have a long history of working together. In fiscal year 2022, the United States Postal Service received over 354 million pieces of mail, international mail. And so that doesn't include things like FedEx or DHL or any of the express courier hubs. This is just talking about the US Postal Service. And as you can imagine, a lot of people try to ship things that they shouldn't through the mail, because with that many pieces of mail, it's very hard to look at all of them. And to that end, the FDA has been trying to not only increase our staffing to look at more of these packages, but to set up satellite laboratories on site to do some analytical testing and hopefully speed up the process so that we're not actually sending so many things to our brick and mortar labs. Also in fiscal year 2022, 71% of the items that were screened by FDA ended up being refused entry, and 56% of those were identified as violative drugs. So there are some scary things coming into our country through the Postal Service. By the end of fiscal year 2023, we had looked at over 63,000 products, and more than 38,000 of them were refused admission, and more than 31,000 of them were violative drug products that ended up being destroyed. So our hope is that we're going to expand the amount of satellite laboratories we have to get those numbers up even higher. So we have 9 international mail facilities scattered throughout the country. And we currently have a full service, not a full service, but we have a satellite lab that is full-time staff with FDA analysts in Chicago. And we just opened up one, it says Doral, Florida, but it's really it's more like Miami. So that one is also open, and we are rotating analysts in from different laboratories right now, but we have started the interview process. If any of you are with the government, you know how long it can take to hire someone. So we're moving as fast as we can with that. Today, I'd like to tell you kind of the evolution of the satellite laboratory. We really started working on this seriously in 2018 when we received a lot of money for the opioid crisis. So much of what we started with in our minds was that we wanted to prevent opioids from entering the country. And it turns out that since Customs and Border Protection, or CBP, gets first pass at all of the parcels, typically anything that looks like a street drug or drug of abuse, they're going to keep that for themselves or else pass it on to DEA. So it turns out that we don't get as many opioids as we were hoping. And because of that, we widened our scope to really look at anything that the FDA regulates. So in terms of pharmaceuticals, we're not actually looking at food or really cosmetics right now. But any type of pharmaceutical product we are attempting to look at out there in the field. So I'm going to tell you a little bit about Operation Snapshot in a couple of slides. And then we're going to talk about the pilot study that happened at the Chicago IMF. We're going to talk about the inaugural year of that satellite laboratory. And then I'll give you some statistics about how Miami is going so far. Back in 2018, we evaluated a number of different portable techniques. So we looked at a combination handheld Raman FTIR, 2 different types of handheld Raman, a handheld mass spectrometer, that's the MX908, a small benchtop FTIR that you can pick up and move very easily, and it just uses a little laptop to power it, a portable ion mobility spectrometer, as well as what we call affectionately the dart on the cart. You can see that here. Yeah, so it is all combined on this cart. We've got the mass spectrometer, which is made by Waters. It's the QDA single quad, but we do get fragmentation information due to in-source collision. It also has the DART ionization source. The controller box is up underneath the cart, and then all of the pumps and the computer are on there as well. You can get small gas cylinders because we do have to use high purity nitrogen for the system, but you can get small enough cylinders that they'll actually fit under the cart. And so we did actually move this around throughout the international mail facility. When you plug it into just a regular wall outlet, it takes about 10 minutes for the mass spec to pump down. So, you know, you can move from place to place pretty easily and be up and running quickly. Operation Snapshot took place in 2018 as well. And during this, we kind of call them a blitz, we sent 8 FCC analysts out to 8 of the international mail facilities. The only one we did not go to was the Virgin Islands. We were working closely with our consumer safety officers, or CSOs, as well as 16 of our Office of Criminal Investigations Special Agencies or agents, or OCI agents. And those are our FDA people that are actually cops. So they have guns and badges, and they get to go on search warrants and pretty cool stuff. So they were helping us out during this split. So during the week, we looked at thousands of samples, and then we chose 85 to send back to Cincinnati at the Forensic Chemistry Center to look at all of the things that we were considering for our toolkit, as well as doing confirmatory analysis using either LCMS, CCMS, or a benchtop FTIR. So based on that information is how we chose the toolkit. And I will say that during Operation Snapshot, I rented a U-Haul and pushed the dart in the cart into the back and drove it to Chicago. And it was pretty exciting, and it was very mobile, even though it's obviously not as mobile as the handheld devices that we also use. Based on the data that we obtained from Operation Snapshot, we found that if we got either a match from the handheld Raman, we went with the Progeny by Rugaku, or the IS-5 FTIR, as well as the same match from the mass spectrometer, the DART MS. It was 98% as effective as what we would see in our laboratory using, the traditional techniques that you would find in a brick and mortar lab. And so that's very important because we have this orthogonality for our toolkit. If we get a match by both the Raman and the FTIR, but not the mass spec, we call it verified. And then that's kind of a judgment call on if we're going to send it to the lab or not. If we get a match from only one of these devices, we just call that detected, and we most likely will send that on for further testing. But if we get, as I mentioned, a hit from either the Raman and the FTIR and the DART MS, we can just stop it right there. And in many cases, we can even destroy it right there. So that's a huge improvement on the way we were doing things at the IMS. So looking at our toolkit, the DART MS is minimally destructive. With the sampling, we typically will take a tablet, cut it in half with a razor blade, and rub that broken edge on a swab. You can see the swabs down there in the bottom. They're the same type of swabs that are used at the airport to rub your hands or your suitcase to look for gunpowder residue or those types of things. If we have a capsule, we empty the capsule shell contents onto a clean piece of whey paper, and we'll press the swab into the powder. And if we have a liquid, we just pipette it onto the swab and allow it to dry. So we do have this thermal desorption unit that's in between the DART source and the mass spectrometer. And that was really important because, as I mentioned earlier, we started this project because of the opioid crisis. And so we were really concerned that our analysts might be exposed to fentanyl or things that are equally as toxic. And so it was really important to us to have this enclosed sampling capability so that they weren't being exposed to aerosolized fentanyl or things of that nature. Now, we don't see a lot of fentanyl out in the field, but it's good to know that we're being safe. The analysis time, including sample prep and the run time, is about 1 to 2 minutes. So our method itself is 20 seconds. And then we use a software called Tamisa that does data matching, and that can take 30, 60 seconds as well. It is quite sensitive. We can get down to low nanogram quantities, and we do sometimes see multiple actives even without the front end capability of separation. So that's been a nice benefit as well. However, it does have a much larger footprint than the other handheld devices that we're using. So if we are deploying someone out to, say, a DHL or a UPS center where we don't have a satellite lab established, we can't always, rent the U-Haul and take this guy with us. So we're always looking for smaller and smaller mass specs if we can find them. And it does require high purity nitrogen. That's another kind of drawback to using this particular instrument. Out in the satellite labs, we have nitrogen generators, which has been so much nicer than swapping out cylinders. So that's been a huge help. Okay, here are some photos of the Chicago International Mail Facility. As I mentioned earlier, Customs and Border Protection takes first pass, and they look at all the parcels by X-ray. If they see something of interest to them, they're going to keep that. They might pass it on to DEA. But if they see something that looks like it's an FDA regulated product, they put them in these big orange bins. And sometimes we have 8 to 10 bins just overflowing with packages. And then our consumer safety officers will open those packages, they take photos, they document everything to the letter. And if they see something that looks problematic or somewhat suspicious, they turn it over to our analysts in the satellite laboratory. So you can see in the top right corner, that is actually the laboratory. It's in a trailer outside of the International Mail Facility. And here's what it looks like inside. So we do have a hood. We have a small amount of bench space. We do share this space with CBP, and they have a full-size GCMS in there. We don't share our instruments, but we do share the space. Okay, so we base our kind of validation of this method on the pilot. The pilot was conducted for 68 working days from 2019 to 2020. We got shut down a little prematurely because of the pandemic. At the time, we were flying people from all of our labs all over the country to come and do these 30-day details. And of course, that wasn't going to be safe back then. So we did stop after 68 working days. But when we looked at all of the data, we felt like we had plenty to say that this is a program that is worth supporting. So we looked at 926 individual items, and some of the items did not have any labeling, so nothing was declared on there. They were just loose tablets or loose capsules and bags. But there were 812 items that did have things declared. And in some cases, there were multiple APIs declared for that product. So this first slide is showing us that of the items that had declared APIs, in 90% of the cases, at least one labeled API was detected by at least one of the techniques. So either the Raman, the FTIR, or the Darnamet. In 78% of the cases, at least one labeled API was detected by at least two of the techniques. And then in about 10%, we didn't detect any of the APIs that were on the label. Now, we're not calling this a true false negative, because I think, as we know, sometimes things do not contain what they are labeled to contain, or they contain something different, or they don't contain anything at all. So this is just kind of looking at the types of things that we were seeing in Chicago during the pilot. Okay, we did send 127 items back to the Forensic Chemistry Center for confirmation. And of those, 90% of those products, we got an exact match by either using LCMS, GCMS, or a benchtop FTIR. So we got an exact match to what we had seen in the toolkit. In 10% of the cases, we got a partial match. So that means that either only one of the APIs that we saw in one of the labs was seen by the toolkit, or maybe we got the same answer, but then we also maybe saw a false positive with the toolkit, because that does happen occasionally. But in 0 cases, we had no matches. So that was very, very good news for us in terms of keeping this program going. So for the validation, we had 672 of the APIs that we detected during the pilot were detected by this technique, by DART-MS. So that's 72%. At the time, we only had one instrument, and it was out in the field. So whenever we encountered an API that was not in the FMISA database, we would go and use things like MC Cloud, PubChem. We would look at literature. We would look at historic data from our lab in Cincinnati to make the matches manually. So we were using 3 structurally significant ions to say that it was really there. However, eventually we bought a second instrument. So then we had one at the FTC and one in Chicago. And we added all of those standards that we could to the database, and then we reprocessed that data. So looking at that is kind of pointing to the ruggedness of this particular method. So during the pilot, we had 13 different analysts. Some of them had never done mass spectrometry at all, and none of them had ever used a DART. So they all came to the Forensic Chemistry Center for a 1-week training course, which was really remarkable, because during that 1 week, we taught these 13 analysts how to use the FTIR, the Progeny, the DART-MS. They learned sample prep, they learned extraction techniques, They learned how to write an abbreviated worksheet. They learned how to troubleshoot. They learned how to do maintenance. And they learned how to take things apart and put it back together, all in 1 week. And then they all went out and had very successful details for 30 days at a time in Chicago. So using our second system, as I mentioned, we were able to add 108 standards to the database, and there were 31 standards that were not amenable to this technique. So we just didn't get a clean enough spectrum to add that to the database and feel confident that it would make a good match. So this is just looking at the true false positive and false negative rates from that validation study. So after adding the 108 compounds to the database and then going back and reprocessing it, we found that 90% of the reprocessed files gave us true positives. We did find that 24% of the reprocessed files gave us false positives. So that means that we were getting the correct API, but we were also getting some other things that were not actually present. So we do often, especially as the databases get bigger and bigger, we have isomers that have the same chemical formula. We have things that have similar fragmentation patterns. So a lot of times it's a judgment call of looking at all of the things that it's saying as a match and really looking at the spectra and deciding if it's there or not. And of course, we're always relying on the toolkit as well to see if we got a match using the FTIR, the progeny Raman. We did find that 10% of the files we got a false negative. And again, 31 of the compounds that we tried to add to the database, we just could not get a clean enough spectrum using the experimental conditions of this method. And we are being careful. We don't want to be modifying methods out in the field. Even within the FCC, we're kind of sticking with the same method just because it seems to work well for most compounds that we see in the field. And we just kind of want to keep everything the same. Okay, we also looked at minimum detectable levels, and you'll notice for these selected compounds, it kind of ranges. We went as low as 10 nanograms and as high as 500 nanograms. We did a lot of work with fentanyl and other opioids and fentanyl analogs. And for many of those, we could get down as low as 5, and we didn't try going lower, but we probably could have gone lower than that. And these experiments were done just by making standard solutions of a known concentration and then adding a known amount to the swab, allowing it to dry. And they were all collected in triplicate. I apologize, it's kind of hard to see these numbers, but what I wanted to just point out from here is these are the laboratory analytics collected through the middle of September. So it's not completely up to date. But you'll see that while most of the things that we get are unscheduled because CBP and DEA take most of those drugs of abuse, but occasionally something will look like a pharmaceutical product and it turns out it's counterfeit and it does contain scheduled substances. So you can see that is listed here, as well as unapproved products. We see those kind of often, and that can be anything from foreign market or just a drug that has not been approved by the FDA at this point. 801U is a list that contains potentially harmful compounds that we have destruction authority for. So if we find an 801U compound and we get it verified or definitely confirmed, but sometimes even verified, we can destroy it, no questions asked. So we're always kind of trying to add more things to the 801U list to help speed up our process of protecting the public health. But one other thing I'd like to point out is in the second column, that's where we're looking at kind of lot units. So if you think about 593 items, that's not necessarily that many. But if you look at the number of tablets and capsules that were in that particular package, it could be thousands and thousands. So in that case, it is making a significant impact on, preventing things from entering commerce that could potentially be harmful. And this is kind of showing us as well what happened to those things. So we can destroy it. That's the best option, because if we refuse it and it gets sent back to the sender, sometimes they'll just send it even back to the same IMF, but a lot of times they'll send it to a different IMF and try to get in that way because they're very savvy to the fact that we can't look at every single package. OGA means it goes to another government agency. Release, it goes on to its final destination, and then we had some that were pending. So just this August, we opened the International Mail Facility at the Miami, in the Miami IMF. And this one is a little different. Instead of being in a trailer, it's actually a room inside of the IMF. And that's so much nicer because our analysts don't have to walk outside whenever they need to go to the restroom or get a drink or any of that kind of stuff. They're right there. And it also really helps with kind of the morale and the social aspect of working with the consumer safety officers, because our analysts can actually just pop out there and see what people are opening and say, hey, that looks suspicious, or we've seen something like that in our lab before. So it's really a nice situation. And so again, this is only going through 9/13, but I believe when I was checking this morning, we're up to just over 100 samples since we opened in the middle of August. So they're keeping us pretty busy down there. These are the top 10 APIs encountered by occurrence. So this is a combination of both Chicago and Miami. And so we see, strangely, a lot of fluoxetine, which is an antidepressant. Not surprisingly, we see a lot of sildenafil as well as other erectile dysfunction drugs. But you'll notice that the biggest piece of the pie just says others. So we see a significant amount of just one-off, odd types of drugs coming through. And so again, that's why it's really important that we have a database that we can add to, because we're constantly updating it based on what we see in the field. And typically, if someone in the field reaches out and says, hey, I think we are seeing this, but it's not in the FEMISA database, if we have that standard in-house, we can turn it around within 24 hours and send them an updated library. Okay, so apologize, this looks a little blurry. But this is showing us the top APIs in terms of lot units. So again, that's pointing out that it's not always the individual item, it's how big that package is to know what kind of impact we're making. Okay, so in conclusion, almost 80% of the unknown, unlabeled products that we've analyzed in the field have been found to contain APIs. And the satellite lab has helped keep out more than 500,000 potentially dangerous products from reaching the American public. And this is just my little plug. We're always constantly recommending that we can expand the 801U list to include each unique API that we detect at a satellite lab using at least 2 techniques, so that we can destroy those things on site and not have to worry that it's going to go back to the sender and then come back through, and we're all just wasting our time. So again, just some acknowledgements. We've been working on this, as I mentioned, for 5 years, and some of the analysts that were crucial in starting this out have moved on to different jobs. So I just wanted to include them. And then I wanted to show our team, and we took probably 10 pictures before we got it where everybody was smiling and nobody's eyes were closed. So I really love this photo, and I'd be happy to answer any questions.
Awesome. Thank you so much, Sarah. Really interesting stuff on a kind of unique application of forensic science and dart analysis. Quick question for you. How did you decide to use nitrogen over helium, and how are you finding using a source gas that not most people use?
Yeah, that's a good question, because previously I'd always used helium, especially with the Orbitraps and those types of high-res mass spec. But when we were first pitched the idea for this dart on a cart, we were concerned about, well, what are we going to do about the helium tanks and how are we going to get those out in the field? And IonSense actually said that they had been doing a significant amount of work and found that nitrogen worked well with that system and gave the same amount of sensitivity. So we started right out the gate using nitrogen, and we haven't had any problems other than at the very beginning, we were getting these small cylinders delivered to us, and they ended up being pretty low purity. And so you could tell right away when you would hook up a new tank, if it was not high purity, you wouldn't hardly see any ions. So once we went to an actual generator, we've had no issues whatsoever.
That's awesome. Cool. Thank you.
Sure.
We had some questions come in, so we'll go ahead and move over to those. One question that came in for Sarah: is FDA using DART with Raman or IR as confirmatory, and are there problems with destruction with regards to legal repercussions?
Yes, so we are using, if we get a match by either the FTIR in the Raman and the mass spec, we call that confirmed, and we are able to take action on that, legal action. So we have all that data from the pilot, and now we have a whole year data set, because we do still periodically send a certain amount, it's maybe like 10% of the samples still get sent to FCC to just make sure that we're seeing the right stuff. But when we work with our consumer safety officers, that's working with our compliance branch, and they have giant manuals of what we're allowed to do and not do. But one thing to keep in mind is technically you're not supposed to ship drugs through the mail, and you're certainly not supposed to ship like loose tablets or loose capsules and completely unlabeled things, and you're not supposed to order stuff from like India or China. So, in a lot of cases, even without our analytical information, we can take action on some of those things. So this is just a little bit of extra information to have to back it up. And nobody that I know, we have not had to go testify in terms of the satellite lab yet, but as an analyst at the Forensic Chemistry Center, we certainly have to be expert witnesses pretty often.
Awesome. And then what type of analyses are you doing when it goes to the brick and mortar lab?
We typically just do LCMS. We were starting with GCMS, but the mass range is so much more limited that we found that we were almost always having to do LCMS as well. So we've just kind of gone straight to LCMS first.
Awesome, thanks.
Sure.
Sandra, there's a question for you. Is DART on-site training reserved for DEA, or can other agencies request training?
Well, at this point, this is customized training for not only the standardized instrumentation as well as the standardized method, and pretty customized based to fulfill our internal, policy and procedures. Yeah.
Awesome. And then I guess another question. At one point you mentioned category A. Can you clarify like when you're using this for category A versus category B, or only category A?
So in terms, and I guess I can answer that from the sweet drug point of view. So since, category A is reserved for when you actually obtain selectivity as high to obtain structural information. So it's not until you get to the fragmentation pattern and identifying moieties in the that it would be considered a category A. So otherwise it's not being used to the highest selectivity that the technique could offer. Yeah.
And then one question we asked both of the speakers last week as they talked about implementing DART in their laboratory systems is, what is one key takeaway or one thing you learned that you weren't expecting as you went through your implementation validation processes? Or one thing that surprised you thought it could do, couldn't do, or you didn't think it could do it and it could do it?
Well, in terms, so I guess I was surprised about one of the thermally labeled techniques we've had some exhibits on catasolam, and that's a limitation because it's thermally labeled and all you see is diazepam on the GCMS. And I was surprised that catasolam was able to survive actually the 250 degrees of the source that we're using. We're studying lower temperatures for that, but you can see a clear molecular ion for the intact catasolam species. So that was good. So it's a limit, something that we cannot do with any other, short of doing LCMS and a longer method. So yeah, so that was good.
Yeah, I think for me, I was really surprised. I was very skeptical when I saw the video that IonSense created way back in 2017, 2018, where they're pushing the cart out in the wilderness, and I think they had some kind of power supply or something. And I remember thinking that's never going to work. But then when I put it in a U-Haul and took it to a filthy warehouse in Chicago and it worked, I was really shocked. And we still have that same instrument, and it's run at this point more than 2,000 samples. So it's really a workhorse.
Awesome. Cool. And then do either of you use negative mode, or everything that you guys are working on is positive mode only?
We've been sticking with positive.
Yeah, we are using positive mode so far. There are some non-controls that we had done, for example, like negative ion mode electrospray in the past, that we may, especially there's one that's a thermally labeled species that we want to try negative mode, but at the moment we're just on positive mode. So.
And I guess maybe more for Sandra, but what is the time it takes to switch between LC and DART?
Without counting, the actual testing of the performance verification that you must do after, it is about 2 to 3 minutes.
Yeah, that's pretty quick. And then I personally have one more question of, when you guys go to do libraries and you're building your own libraries and databases, what type of curation are you doing? Are you training people how to make sure that the spectra is logical before they accept it, or?
We try to just observation in terms of expected. And we're only isolating the monoisotopic mass, not the killing the C13. So kind of curated for that, and now having the NIST MSMS libraries available, it's a huge help, because the fact that we can create the same spectrum they create, and sometimes in a different mass analyzer. Sure. So we know what to expect and what may be extraneous or not. And if our compares positively to the NIST one, and it's a high quality one, then it's good for a library. So.
Yeah, I was kind of laughing when you brought this up, because we just had an issue where we had created a library entry and there was some carryover, but the person who created the entry didn't notice that, and so they sent it and we were not getting matches. So we just recently started a new thing where another analyst looks at it as well and compares it to either NC Cloud or historical data. Even looking at LCMS data is usually very similar. So we are now getting another pair of eyes on that, because it could easily happen.
Yeah, it's a difficult thing to have to control for sure.
Yeah, especially like for folks that are not familiar, and that's why we're not letting everybody enter data into the libraries, and we'll have usually the Q group, especially in terms of what people get used to, what's the normal noise level, what's the normal level for good signal or not good enough signal, especially since as you break the molecule apart, that signal gets distributed over all the fragments, so you start seeing more artifacts at those low levels. So yeah, so an extra set of eyes is always good.
100% agree. I guess just to be cognizant of everybody's time, we're coming right up against the 3:30 mark, so we can go ahead and close out. Again, we'd like to thank both Sandra and Sarah for taking their time today to come and talk to us about how both the DEA and the FDA are using DART Mass Spec for their analyses. So thank you both again so much for taking the time to speak with us.
Thanks, everyone.
Thank you.